Hello everyone, thank you for joining us today on this exciting journey through the intricacies of genetic engineering and the captivating world of transcriptomics. I'm Nishad Mohammed, and it's my utmost pleasure to introduce my co-presenters, Kayleen Long and Dakota Jones. In the vast landscape of biomedical research, genetically engineered mice or GEMs stand as powerful allies, offering us a unique perspective into the intricate dance of genes and their contribution to disease. We harness the potential of bioinformatics to decode the secrets within the genome of these remarkable creatures. Our journey is not solitary. We endeavor to reproduce and expand upon the groundbreaking transcriptomic work conducted by Farkas et al. 2019 in their influential article titled Streamlined Computational Pipeline for Genetic Background Characterization of Genetically Engineered Mice based on next-generation sequencing data. Their work provided us with a roadmap and ignited the spark of our exploration. As we navigate through our methods, you will notice the threads of their methodology woven into the fabric of our own transcriptomic pipeline. From this study, we point out that it is critical to grasp the significance of a specific genetic player called CDKN1, also known as P21CIPWAF, and this protein is encoded by the CDKN1A gene. It acts as a critical regulator of the cell cycle, inhibiting the activities of CDKs and orchestrating the progression of cells through different phases. It not only influences the orderly progression of the cell cycle, but also responds to DNA damage and stress. It is often induced in the response to genotoxic stress, playing a pivotal role in mediating cell cycle arrest and allowing cells to repair damaged DNA before proceeding through the cell cycle. We also looked out for these details in our results. In our methods, our journey into the world of genetic exploration started with a meticulous selection of data inspired by the rich datasets utilized by Farkas and their team. The GSE 123-168 dataset is a comprehensive collection of RNA-seq samples, and it became the cornerstone of our study. These samples are categorized into controls and doxorubicin-treated, nulls, and heterogeneous and wild-type groups. And it provided us with a canvas on which to paint the story of the gene expression. Quality control was our first line of defense, leveraging FastQC and multi-QC to scrutinize our raw data and ensuring its integrity, setting the stage for the journey ahead. Read alignment was carried out by HiSat2, sorted and indexed with some tools, and with our align reads in hand, feature count stepped in to quantify the gene expression, providing us with a comprehensive TSV file brimming with counts. Using R, we took the reins to create a count matrix, a structured view of our expression across all samples, echoing the methodology established by Farkas et al. We replaced the edge R, which they used, with DE-seq for our differential expression analysis, which became the focal point of our exploration. This not only validates our findings, but also contributes to the robustness of the scientific knowledge. We used DE-seq2 to create contrast between the genotypes and the treatment groups. As we immerse ourselves in the visual tapestry of our results, inspired by the visualizations utilized by Farkas and their team, bar plots, Manhattan plots, ridge plots, and dot plots, we extract inference that paved the way for a deeper understanding of the genetic intricacies within the genetically engineered mice. In upcoming sections, we'll explore the implications and insights derived from this comprehensive informatic analysis, building upon the foundations laid by Farkas et al. 2019. I'll leave the room for Dakota Jones to take up the results and the discussion sections. Thank you.
We first wanted to start with a visualization to confirm that the data does contain knockouts and ratios as expected compared to wild type. It was a nice sanity check to see that the expression overall for SALL2 was lower in knockout models compared to wild type. We likely could have better visualized this result if we had normalized the sample amount. As is, homozygous has 4 more samples compared to the other groups. We wanted to explore the data in its entirety, so we elected to keep these counts in. Since SALL2 was not a significantly expressed gene in our DE-seq2 analysis, this is the last we saw of it in our pipeline. However, this is in line with the findings of Farkas et al., as they did not find it as a significantly expressed gene in their data as well. We next wanted to explore the results of our DE-seq2 analysis. We elected to use PCA to help determine factors possibly explaining our observations. We found that when accounting for biogroup, PC1 was not likely explained by genotype, as all genotypes were found on both sides of 0 in this plot. It is a possibility that PC2 is explained by genotype as it affects the placement of wild type towards the top of the plot. Next, when accounting for treatment of doxorubicin, PC1 seems to be best explained by treatment. Control groups and treatment groups separate into left and right-sided clustering. We next considered both factors, treatment and biogroup, with the resulting plot that again shows that treatment groups separate seemingly based on PC1. Our next goal of the pipeline was to explore gene set enrichment in our results, specifically in regards to gene ontology. We elected to compare results from an R package named gprofiler2 to results obtained by the cluster profiler package we used in class. gprofiler2 allows for simple exploration of enrichment and visualization of gene ontology results using 2 function calls, ghost and ghostplot. This results in a Manhattan plot that includes a table of top gene ontology terms based on p-value. Next, when looking at wild-type homozygous knockout, we see results that indicate significant enrichments and pathways involved in defense response. This is likely explained by the doxorubicin treatment and is in line with some of the results we saw in the paper. When looking at wild type versus both homozygous and heterozygous, we are seeing differences in expression tied to the treatment of doxorubicin yet again. We are seeing pathways, commonalities here that are involved with defense, typically by some type of inflammatory, such as a viral or bacterial response, which I think is a good indicator given that doxorubicin is an antibacterial treatment. Next, in the context of a design formula that considers only treatment, we saw many more differentially expressed genes. This may indicate that SALL2 genotype is having an effect on exactly how the cells respond to the treatment. Given more time, we would have liked to explore these results more, as there is much filtering that could have been done to maybe find more significant results. We then carried out a similar analysis using the cluster profiler methods we learned in class. We were happy to see that the results for enriched pathways were very similar to our gprofiler2 results, indicating its usefulness in this type of analysis. Again, we are seeing defense response pathways enriched in this analysis of wild type versus homozygous and heterozygous. We were also seeing similar pathway enrichment results between gprofiler2 and cluster profiler for control versus treatment groups. Finally, we included KEGG pathway visualization. This time we used gprofiler2's gconvert function to quickly convert to interest gene IDs for use with enriched KEGG function. We pulled pathway visuals for the top results of enriched KEGG. To the right, you can see the top KEGG pathway enrichment for the wild type versus homozygous knockout. We included this to show that you can pull these visuals using our script, but we won't take any time to explain what is going on with this pathway. For the most part, we were able to successfully produce results that are in line with some of the results found in the paper, barring the fact that we did not carry out any variant calling analysis. Because of this, we do not have any variant calling results to compare to. We found that the SALL2 knockout mice had suppressed CDKN1A expression, as it did not appear as a significant result in either differential expression analysis or in gene set enrichment analysis. Finally, we are happy to see that the G-profiler 2 and cluster profiler pathway results were very much in agreement with each other. Given this, we could recommend G-profiler 2 for a quick and dirty gene ontology analysis using gene sets. As shown earlier in our presentation, a GitHub repository link is available where you can find all scripts used and all FastQC and multi-QC results.
In terms of a retrospective analysis, we did end up making some changes to the original pipeline. First off, the paper provided 2 implementations of the pipeline, the web-based version using the Galaxy platform and a Bash and R scripted form. The Galaxy platform does come with some difficulties pertaining to the availability of computational resources, however. As a result, we chose to carry out the Bash and R scripted version of the pipeline. This meant that we were not able to evaluate the efficacy of the pipeline using the Galaxy platform. We also faced some difficulties with the data chosen. The sample dataset generated by the paper was an RNA-seq dataset focused on evaluating variants caused by doxorubicin treatment. As a result, a lot of the differentially expressed genes and the differences detected by the pipeline were results of these variances in treatment. We had some difficulty in accounting for these variances enough to detect differential expression solely due to genotype. Incorporation of an untreated dataset might have allowed us to more accurately assess the pipeline's ability to detect genetic variation between samples. This generated dataset also featured a relatively low number of samples, making for reduced confidence in our results. We also made some adjustments to the packages used within the pipeline. This paper was published in 2019, and the EdgeRun R package was used for differential expression. This package has since been removed from CRAN for various issues, so we chose to use the DE-seq2 package for this portion of the analysis. We made this decision due to DE-seq2's widespread use and documentation and our own familiarity with the package. We additionally tested both G-profiler 2 and cluster profiler for enrichment analysis, eventually settling on G-profiler 2. Finally, we did have to make some changes to the scope of the pipeline due to time restraints in the scope of this course. This pipeline was constructed for use with multiple types of next-generation sequencing data, such as RNA-seq, whole genome sequencing, and whole exome sequencing. We were only able to implement it with RNA sequencing data, however, and so we were not able to evaluate its utility in the application to either whole genome or whole exome sequencing data. Additionally, because of time restraints, we were only able to carry our data set through the differential expression, gene ontology, and KEGG pathway analyses, eliminating the variant calling portion. This was done both because of the time restraints and also because variant calling is more often conducted through the lens of genomics rather than transcriptomics. Overall, we learned a lot from our implementation of this pipeline. We were able to assess the utility of the pipeline for genetic characterization through the detection of differentially expressed genes and enriched pathways. Our implementation focused on the pipeline's application to RNA-seq data that was focused on the effects of treatment, but we were able to detect the effect of treatment on gene expression and identify the SALL2 knockout. Direct observations of differences in singular gene status were accomplished through count analysis of SALL2, and gene-induced differences in pathway enrichment were observed within the context of doxorubicin treatment. This demonstrates the utility of the pipeline for characterization of genotype and its effects in genetically engineered mouse models, either as an in-between step when conducting a breeding program or as characterization before studying the effects of a novel treatment. While we were not able to thoroughly assess this pipeline's utility in all modalities and with all types of data, we did find ways to strengthen it through the use of alternative packages. Specifically, an outdated package was replaced with a more well-documented and current option, and package choices were solidified for each portion of the analysis. As a whole, our analysis of this pipeline confirms its utility in the characterization of genetically engineered mouse models from RNA-seq data, and makes an effort to continue its expansion into the current R ecosystem with the application of new packages.